So welcome. I have a talk tonight on chlorophyll and hemoglobin, and I was noticing that the chemical structures of these two important life molecules are almost the same, except for a couple of differences, and I'll show those to you. But they have two different functions in life, one in plants and one in animals. And so I wanted to introduce that subject to you today and talk about it. My name is Doug Sharp. This is my wife down here, Cookie. Cookie and I just got married about a year and a half ago, and we're newlyweds, and so she's the love of my life, and I appreciate her support and help. And so I'm going to tell you a little bit about what we're going to do today. We have an organization called Revolution Against Evolution. We have a website which is very simple, www.rae.org. And then we have a YouTube channel, www.youtube.com/RAEORG. And our purpose is to answer the tough questions of science and the Bible. We have been doing this TV show since 1993. And so I think this would be episode number 1033. So it's been a long haul that we've been doing this. It's been a lot of fun. Started out teaching in the Mount Hope Bible Training Institute, which I don't think is operating anymore, in Lansing. And I moved here from Lansing to Grand Rapids in 2023. I bought a house next door to my daughter. We rented it out a couple of years, and then I decided to move over here, and a lot of things in my life changed. I met this young lady here, and she's going to be celebrating a birthday on Sunday, so wish her happy birthday. I'm not going to tell you how old she is. 39, yes. I wanted to also tell you that I've written a few books. These are some of the books that I've written. I have a book, my newest book is called Don't Let Your Geology Mess Up Your Theology. And we just came back from Arizona and did a tour of Sedona. Some of our Sedona pictures are in this book because I go out there frequently. My daughter lives out there. And so we talk about areas in the world where the geology does not match what is expected by evolution. You know, where the oldest layer is, which is supposed to be down here, is actually up here, and it's on top of some of these younger layers. And I got a lot of examples of that in my book. And it is a picture book, and it's a lot of fun to read. I am hoping that I can get some reviews for my books online on Amazon, because there's been a couple of evolutionists who don't read my book, but they give 1-star reviews because it's not an evolutionary book. It's openly creationist, so I think that they are just trying to damage me, and so I've got like 4 or 5-star reviews. I got about 5 1-star reviews. So I have a little conspiracy going against me. I am also interested in history. My brother-in-law worked for the Michigan History Center in Lansing, Michigan, part of the state of Michigan. And when my grandmother passed away, I found an old book in her attic, which was written by a doctor. It was written in between 1844 and 1863, and all these handwritten pages I felt was worth preserving. And so what I did was I scanned each of the pages, and on the left page is the scanned version. And then I go into each of the ingredients and define them and figure out what they were, and it's really fascinating. A lot of the things that they had discovered worked for curing things, and we use some of this stuff today, and then some of the stuff, if you actually tried it, you'd probably get put in jail. I'm not sure I actually recommend the remedies that Dr. Hale put in this, but it was an interesting study because I wondered, well, who was this guy and where did he come from? I did an internet search on the Hale family, the names in the book, and I found a 1913 reference of a old history of Calhoun County, and Dr. Hale's brother was in it. And so I found that Lucy Hale and Jerome Hale were the parents. Wallace Hale was the doctor who wrote the book, but he was using his grandfather's remedies, Benjamin Hale's remedies, and that's what we have in the book. And so I document this, I put this together. It's gotten a lot of attention online, especially from scammers who want to sell me republishing a contract. And I've been dealing with that kind of nonsense. Then this book, No Worries in Heaven. I was married to Vivian for 49 years almost, and she contracted cancer and preceded me in heaven. And I talk a little bit about the process that you go through when you're bereaved, when you have someone suddenly pass away, and you have all these different things you have to take care of. And you don't think about that until the time comes. And so I deal with how I ended up dealing with my wife's passing away and taking care of all the, and I sort of detail all the points of, you need to have a death certificate, you need to have each of these problems that do come up, and it's overwhelming when you have a spouse pass away. So I think it's helpful for someone who's going through that. My first book is called Revolution Against Evolution. I published that in 1986, the first edition, and I had 2,500 copies of that printed. Then I did a second version of that with 5,000 copies, and I don't have any more of those left. And so this is the 5th edition. I kept revising and kept bringing it up to date. A lot of it has to do with the top points of evolution that really don't work. And so I identify these things, and it talks about geology and common arguments, all these different things, early man and mutations, origin models, stars and planets, creation models, thermodynamics. You know, a lot of these things are standard things that you are probably all familiar with. But this is a good book to give to a beginner who really wants to know what the creation evolution issue is all about. I had 2 different books that were commissioned by the Creation Research Society. There's Persuaded by the Evidence and Transformed by the Evidence. And Persuaded by the Evidence, there's actually, I think there's like 52 chapters written by scientists, and their testimonies of what evidence in science caused them to go from evolution to creation. And Transformed by the Evidence is a little bit more in depth, showing what actually changed in the person's life when they came to realize that the evidence shows that there was a creator, how it changed their life. I only have one copy of that left, but it's out of print. The publisher went defunct. And so that's another problem with the publishing, is that if you get a publisher, you have to make sure that they're going to be around for a while. I self-published my own family history, the Sharp Dairy Farm. And then we've been doing some books for Dr. Jerry Bergman. This is one example, the Methodist Darwin syndrome. And so these are all on the table over here. So let's talk about chlorophyll and hemoglobin. Hemoglobin is in the animal kingdom. You've got way over here on the evolutionary scale, the kingdom of animals and the kingdom of plants, okay? And you have these two different molecules. The hemoglobin fixes oxygen, and it gives it a possibility of taking oxygen out of the air, and it attaches it, and this is a transport mechanism for oxygen. And gets incorporated in our system. Whereas chlorophyll it fixes carbon dioxide. It brings carbon dioxide into the plant, and it becomes part of what it uses to bring life into the plant. And my contention here is that these two compounds are so similar that they had to have the same creator, the same origin, the same mind behind it. And so I'm going to go into a little bit more in depth of how it all works. I'm not going to explain the process of how oxygen is incorporated in the body, or the carbon dioxide is incorporated in the plant. That's a subject for another miracle. And you guys can just look this stuff up online yourself. But I'm going to point out this fact that these two molecules have very similar functions. And plants produce oxygen and and bring in carbon dioxide into the plant, whereas the hemoglobin will incorporate oxygen into the body and give off carbon dioxide. And so these two mechanisms in our living systems are totally interdependent upon each other. We need the plants to produce oxygen. We need the carbon dioxide given off by us to produce, go into the atmosphere and become used by the plants. And we have all these nuts out there who say that the carbon dioxide is a pollutant, and it's something that causes global warming, and we really are causing a real problem by producing carbon dioxide. Well, really the plants love it, and it's a real contention for me to figure out why people are thinking that way. A little bit differences of the two molecules here. Hemoglobin has an iron molecule at its center, whereas plants have magnesium as the metal in the center. And what this is, there's an ionic bond between the nitrogens here and the metal ion. And then chlorophyll has this long stringy thing on the, it's called a phytol chain. And I'm going to talk a little bit about these bonds that they have here. This produces what's called a hydrophobic chain on the chlorophyll. What it does is it causes it to repel water. It's an oil. It's like oil. And so the oil causes it to bury itself with this phytol chain into the cell membrane. And that's why this is different than the hemoglobin. The hemoglobin doesn't do that. We'll talk about the chemistry of water. We'll talk about the chemistry of life, synthesis of life components, synthesis of chlorophyll and hemoglobin, and taking these chemical pathways, evidence for common designer from two different life kingdoms. So let's talk about the chemistry of water and the chemistry of carbon. This is water. Water is H2O, and it has a characteristic is that the hydrogens are on one side of the oxygen. And if you know a little bit about chemistry, there's orbitals that these do in 3 dimensions, where it has a positive charge over here. These are positive charges, then this is a negative charge. And the negative charge and the positive charge attract each other in what's called the hydrogen bond. This is very important to understand. You could probably write a 400-page book about the chemistry of water. I'm not going to read that book for you, but it is a marvelous compound that God created to hold everything that we have in life. We cannot live without water. And here's some of the characteristics of water causing it to be essential for life. The water molecules have a polar structure, allowing them to dissolve many substances. It has cohesion. Water molecules stick together, enabling surface tension and capillary action. It's the capillary action that draws the nutrients from the ground up into the top of the trees. Water can adhere to other surfaces, facilitating nutrient transport in plants. It has a high specific heat. Water can absorb and retain heat, stabilizing temperatures in organisms and environments. Then there's the density. Ice is less dense than liquid water, and it allows it to float and insulate aquatic life below. So at 4 degrees below zero, water will freeze and then become lighter, actually be able to float on top of the rest of the water. And so without that characteristic, which is unique to water, we wouldn't have life. Water is known as a universal solvent. It's crucial for biochemical reactions. I want to talk about carbon atom. The carbon atom, the orbits of the electrons around the carbon atom are not circular. They actually form a standing wave. And it's these standing waves around the X, Y, Z axis of the carbon atom that cause it to form what's called covalent bonds. And here's the 3 different kinds of covalent bonds that can form. You have a single bond, you have a double bond, or a triple bond. And it's where the orbits of the electrons mesh together between the 2 atoms, and they go back and forth so that they are able to cling the 2 atoms together. Covalent bond is a nice strong bond. The components of life are amino acids and nucleotides. And I wanted to show you amino acids because these mean old acids are are bipolar, and I'll tell you a little bit about that again. But there's the acid part, which has a carbon, 2 oxygens, and a hydrogen. And then there's the amine group, which is a nitrogen, 2 hydrogens, and it attaches to the carbon atom. Now, the nitrogen atom is able to connect to 3 different bonds, where carbon has 4, oxygen has 2, and so one of the things that happens in amino acids is that this hydrogen over here wants to migrate over to this amine group and is attracted by that, and then that forms a negative charge over here and a positive charge over here. And then what really gives the meaning to living systems and proteins are these side chains. And I'll show you, there's 20 amino acids that form proteins. And the side chains are what give the meaning to the protein function. This is the experiment that was done in 1953 by Stanley Miller, and what it did was he said, well, you can make these amino acids by taking water, ammonia, methane, and hydrogen, put them into an atmosphere up here. We have a lightning source that creates the spark, and then there's a mechanism here to run water or cool it off. It collects all the compounds that are made in this process down here in this little trap, and so they are able to extract out the mechanisms for this. What they don't tell you is that if you let it continue back in the boiling water and go back through this, you sort of goof up the experiment, but that's their problem, not mine. And it produced a number of simple amino acids, along with a whole lot of other junk. But these are the 6 amino acids that the Miller-Urey experiment produced. But there are 20 of them. Okay, here's the 20 amino acid structures. The ones with the stars are the ones that Miller-Urey produced. And you can see, in this case, there's only a hydrogen in glycine. You have a CH3 group over here and alanine. You have valine, which has got more of the carbons here. These are oil-like compounds which don't like water. This is another one, leucine, isoleucine. Methionine has a sulfur in it. Now, the sulfur is important for discussion later. Tryptophan and tyrosine, I did some experiments on that when I was many years ago. Phenylalanine and these are all compounds that repel water. And then proline has got actually a cycle chain here. Serine has has another oxygen on the end of here, and so that's a little bit different. Threonine, cysteine, tyrosine, asparagine, glutamine, aspartic acid, glutamic acid, lysine, arginine, and histidine. And so out of the 20 amino acids, you have 6 of them that the experiments that they tout show that life could have evolved by chance were produced by that experiment. And they are the simplest ones. The more complicated ones, like tryptophan and tyrosine, are a little bit more complicated, and they just don't show up. It's something that doesn't get produced by chance. So what I did when I was doing my experiments back in college, I took glycine, alanine, tryptophan, and tyrosine, basically because they were in the laboratory, I was able to use them as part of my experiment. And they came in two different forms. I'll tell you a little bit about that later. But I applied electric spark to these guys in solution, in water solution. It turned out my experiment was not set up right. But I'll tell you why in a minute. When you produce amino acids in the test tube like they did, or in that experiment I just showed you, you get equal mixtures of right-handed and left-handed amino acids. Life only uses the left-handed kind. It doesn't use the right-handed kind. And so if you want to purify all the amino acids that you're going to use in life, you've got a problem because there's no chemical way of doing it. Basically, it's just they are opposite mirror images of each other. And there's a similar problem with sugars and lipids. Sugars in life, sugars are right-handed, not left-handed. And in life, fatty acids are cis, not trans. Now, you've probably heard these terms lately. But trans means that it's in this configuration like this, whereas cis has a bend in it, and these are higher energy compounds, which means that if you form these in the laboratory, you're going to get a whole lot more of this than you are of this. And so what you have here is oleic acid. Stearic acid is on the top, oleic acid on the bottom. And the oleic acid has a double bond here, and it's the two different side chains here are on the different sides here. Whereas here's the double bond, but it's a different conformation. These are difficult to separate, and they're something that the origin of life people have a lot of problems trying to define and figure out why it is this way. Proteins, when they're put together into a chain, they form an alpha helix. And I'll show you a little bit about that later. But the alpha helix it gives a three-dimensional function for the protein. It folds it up into a ball, and the weak bonds that hold it together can come apart like a zipper and come back together. And so it's that conformation in life that gives it its function. It breaks down your food particles, and whatever you eat is all taken care of by enzymes, which are proteins. Now there are different kinds of weak bonds. You have covalent bonds, which I showed you with the carbon atoms. The covalent bonds also connect to oxygen and sulfur. And then there's ionic bonds, which is like salt. Salt is a negative charge and a positive charge, and it's the attraction of the two charged ions that creates the bond. Hydrogen bonds are a little bit weaker. Then you have the hydrophobic and hydrophilic bonds, where you have either water-loving or water-hating molecules on the side chains of the amino acids. And then there's another one which I have yet somebody to explain to me how they work. It's called the van der Waals bond. And it's very weak, but it is significant because it helps to put things. It's like the sticky tape, when you have little stickers that you put on, is the weak glue that holds things together, and it has a function. So here's what I did in my experiment. I wanted to produce a peptide bond. Peptide bond is where you have an amino acid right here, and you want to combine it with another amino acid. What you do, you have to take out a water molecule, and then the nitrogen over here connects to the carbon, and it forms this peptide bond right here. Now, because I had my amino acids in solution, I was really compromising my experiment because I had the water molecule already out there, and I had no mechanism to remove the water molecule, and so my experiment didn't work. Now, I thought I really had something when my tryptophan and tyrosine experiments were turning brown. And basically what I was doing was I was burning the amino acids, producing a brown sticky goo. When I published an article about this years ago, I called it Interdependence in Macromolecule Synthesis, Evidence for Design. Now, if I were to publish that article today, I'd probably call it Zapping Amino Acids Produces a Lot of Brown Sticky Goo. So anyway, going back to the amino acid structures, each of these side chains produce the different, you have the nonpolar side chains, which don't like water. The polar side chains do like water. Sulfur bridges, where the sulfurs in the amino acids that weren't produced by Miller-Urey will combine together to connect different things in the protein. You have the hydrogen bonds, the ionic bonds, and then these bulky things like this, these side chains with the aromatic rings, they sort of give the shape to a protein. And it's this shape that causes it to have activity in the body. Now, amino acids are zwitterions, and the zwitterions are where you have one end of it negatively charged, and the other part positively charged. And this is the three-dimensional structure of insulin, which is supposedly a simple enzyme. Now, what it shows, you see the alpha helix here, and then you see it's bending around in different conformations. And what it does is that there's this is the compound it's working with, and it's holding it in a position where it can bend it out and break it apart, and so it produces these subcomponents that are breaking down the sugar molecules. So I think that's what it's doing in insulin. And so these things are quite complicated. And one of the things about these enzymes is that if you get enough of these things made where it has a high enough concentration, it'll bind somewhere else on this thing, and it'll shut off the reaction. And so it is a catalyst, and it causes things to break down and work in the body. And so this is the sequence of the amino acids in insulin. I once had a class on this, and I was telling the students that they had to memorize this, and next week only 2 students showed up. So that's a little bit about proteins and amino acids. I'm also going to talk about the DNA molecule and the five components of the DNA molecule. The nucleic acids have adenine, guanine, cytosine, thymine, and uracil. Thymine is only found in DNA. Uracil is its component in RNA. And what you have happen here is you have to have a synthesis process to put these things together. And so there is a whole bunch of steps here that they go through to make this in the laboratory. You choose the desired nucleic acid type. You select appropriate starting materials. You utilize solid phase synthesis techniques for oligonucleotide production, okay. Employ polymerase chain reactions for amplifying DNA sequences. Use reverse transcription for synthesizing RNA from DNA. And purify the synthesized nucleic acid using gel electrophoresis or chromatography. Anybody know what those things are? Electrophoresis? Okay. And chromatography. Well, chromatography, I use this in my experiment to separate all the burnt amino acids in my experiment. There's a column that has this resin in it, and you pour the mixture into this column, and it sort of trickles down this column, and because of the different weights and characteristics of the molecules, they separate. And so you have these little test tubes going along here, and it separates out. That's column chromatography. You can also do that with paper. Paper will do that. And the electrophoresis, you have electrical charge on the same thing, or you can have a grid based upon the electrical charge, and it separates according to its behavior in this medium that is, and these are techniques that you use in the laboratory to figure out what it's like. Now I'm not go into this a lot, but this is actually something that's very important to me. Because I had my experiments in water, it wasn't doing any reactions to form a peptide bond. And so there was a character who, and I forget his name, but he won the Nobel Prize for doing this. He used a process called solid phase synthesis. And so instead of having the amino acids in solution, and there's also something that is necessary for making nucleotides, you have a resin that you put these amino acids on, and they first have to protect one side of the molecule, and they slice off the mean group and put on this protected molecule. And so one of them is called fluoromethyloxyl carbonyl group, and T-butyl carbonyl group is what you do on the other end. And so you have to protect both ends of the amino acids so that when you do the bond, it'll connect at the right spot, otherwise it'll reverse it. And so it's a real involved and low yield process. And so you might get a small polypeptide doing this. So we're going back here, this is what has happened. They take this amine group and put this other molecule on it. Take this off to put the other molecule on it. Let's do the same thing over here on the other side. And then you can bind it right in the right sequence. And so this is what it looks like. This is the one protecting molecule. This is the other one. And so they are able to put that together like that. Now, one of the problems that you have is that if you're making something in the laboratory and making a polypeptide or a protein, this is DNA polymerase, which is a huge molecule. And this is the molecule that is used to create DNA. Now, this is the problem. DNA makes RNA. RNA makes proteins. Proteins make DNA polymerase, which is used to make DNA. So which came first? But the problem is, if you're having this little manufacturing process that you're making come out with a protein, you have 51 amino acids like insulin or something like that, they don't necessarily fold up the way they're supposed to when they come off that little process. And so in life, it's all automatic. It's part of the manufacturing process that life produces all this stuff. And so you have all this folding up that goes on as part of the designed process that God made. DNA polymerase is really a complicated process. It keeps the double helix from kinking, and it manufactures it in the right sequence. It goes through this little hole and it comes out the proper way. If you have any kinks in or anything out of sequence in this process, you're not going to have this work. So I show this. This is here's your DNA template. It peels off a transfer RNA component here, which makes part of a little manufacturing process called the ribosome. Transfer RNA is it gloms onto an amino acid, and it puts it into ready to be added to a growing polypeptide chain. Then you have messenger RNA, which contains the code to make the protein. And so we have this growing polypeptide coming out of this manufacturing process. Then you have that completed enzyme, and it shuts off when it comes to the end of it. And so then this completed enzyme is used, one of them is used as the DNA polymerase. DNA polymerase is needed to produce the DNA molecule. Now this fellow, his name is Dr. Joel Brown. We visited him a few days ago in Arizona. He is the director of the Van Andel Creation Research Center, and that's at the campus of Arizona Christian University. And one of the things Cookie and I decided to do last year was to donate the funds for a DNA sequencing machine. And what you see in his hand right here is one of the DNA sequencing machines. There's actually, you put the sample in here, and there's pores in this, microscopic pores, that is the size of the DNA molecule. And each nucleotide has a charge, a different charge. And this nucleotide sucks the DNA molecule through it, and as it does it, it sequences it, and the results show up on the computer. Here's his supercomputer here. And one of our earlier donations is this machine. It's called the infrared spectrophotometer. That's also used for identifying compounds. That's the machine I used to figure out that I was getting burnt amino acids years ago. So I'm living vicariously through the Creation Research Society, because after I ended up graduating from college and having to change my major because I goofed up an experiment, and ended up in the Navy, I really didn't end up in chemistry. I ended up in computer science. But one of the things the Creation Research Society is doing is extracting DNA from dinosaur bones. And they're attempting to identify dinosaur DNA. If you have dinosaur DNA, that means it can't be that old. Because if it lasts, it could last 1,000 years, thousands of years, but not millions, 65 million years. And so the question is, when you, here they have a dinosaur tooth. And so they're extracting the DNA out of the dinosaur tooth in this project. And the question is, how do you tell the DNA from contaminant? And what you might have is somebody's lunch that you're analyzing the DNA from. Or if they touched the tooth, you might have a little bit of your sweat, and you might have your DNA being analyzed here, not the dinosaur DNA. And so what they're doing is that they analyze all these different possibilities of what might be a contaminant, and then anything that's different than that might be your dinosaur DNA. They figure this might be a 5-year project in order to establish that we have dinosaur DNA. Now they've already established dinosaur soft tissue. They've got some examples of that. And here's some of their laboratory equipment. They're actually cooking some of the bones that they have. They have ostrich bones, alligator bones, cow bones, and then they got some of the actual dinosaur bones. And if you have unfossilized bones of dinosaurs, that's also an indication that it can't be that millions of years old. So this is the equipment that they have. You insert that little device in here, connects to the computer, and it sucks the DNA through, and that's what it creates. If you have a human genome, there's like 6 billion base pairs. So that's a lot of information. Let's get back to chlorophyll. You have the phytol tail, which I told you about. It connects to the cell membrane, and you have a magnesium in the middle. Here you have hemoglobin, but you have the iron in the middle. You don't need the phytol tail because you're not connecting to the cell wall. Here you have the two different molecules compared side by side. Differences being the metal component in the middle. This is, and we don't have to get into this because it's complicated, but this is the formation of hemoglobin. And you have, and basically I looked these up in two different references. And so you find that up to the insertion of iron, the process is about exactly the same. So this is the formation of chlorophyll. It has this same pathway, chemical pathway, up to the formation of the insertion of magnesium. And then you have some additional reactions in the chlorophyll process to produce the phytol chain. And so the phytol chain is this guy here. And that's what attaches to the cell membrane. So my conclusion is in living systems, the manufacture of chlorophyll in plants, similar to the manufacture of hemoglobin in animals. This is up to the point of the insertion of the iron or the magnesium ion and the creation of the phytosanitary chain. I'm claiming that this is strong evidence that these systems were produced by the same designer, that reused the same process, modified it for a different function. And so I say that understanding all of these principles, a biochemist should be the first one in church, first one to use his or her skill to serve God. We realize that our biochemistry is a computer code in 4 dimensions. Its sophistication is way beyond our understanding, and attempts to emulate it or recreate it are futile. So that's my talk. I'd like to give you some chance to ask questions. Yes?
It's not related to the topic, but you said they are cooking bones in that lab that you visited.
Yeah, they want to extract DNA out of them, all these different types of animals, and they're for different purposes.
Can DNA survive being cooked?
Yes.
Oh.
Well, what it's actually doing is it's an extraction process,
Okay.
but it needs to be heated up a little bit to do that.
Okay.
And so another thing that they're doing at the lab is they're analyzing things that are supposed to be convergent evolution. That is where you have like agave that's in the new world and aloes that is in the old world. They're very similar in morphology and structure, and yet they are supposed to be totally different in terms of origin, because they're so separated. And so they say, the evolutionists tell us that it's convergent evolution. Well, if you analyze it, one of their experiments they're going to work on is to analyze those 2 different kinds of plants and see if there's similarities in the design of the DNA in both plants. And so that really is something that is helpful in understanding our origins and finding out if the lie of evolution is false. Anybody else? Well, I thank you for coming tonight.
Thank you, Doug. I really appreciate that. I suspect that many of you were in the same boat I was, and that you weren't understanding everything that was talked about. I think by watching something like this, we can very much verify that things in nature are much more complicated than what could come about by an unguided, mindless process. I wrote a song one time about our relationship with God and how He's living on a much higher plane than we are. And it said, so I pray now, Lord, please teach me to understand your ways, to comprehend the rich design that you have put in place. But I'm also bound by limits, and your ways are way beyond man's. So when I cannot grasp some truth, help me still to grasp your hand. So even though we don't understand everything about the creation that God has put in place, we can still trust Him and know Him, and certainly determine from a presentation like this that this could only be here by the hand of a genius and creative God who loves us very much. Let's pray and we'll finish. We've got food left, so if you want to stay around and fellowship for a while, feel free to do that. Lord, we thank you for this presentation. Thank you, Lord, for these amazing processes that you have put in place that support our bodies, support plant life, support the whole of creation. It's the very foundation of the food chain where chlorophyll is operating, and you put an amazing design in place there in the process of photosynthesis to take photons from the sun and turn them into sugar molecules that we can eat. And that's an amazing thing. We thank you for that, and thank you for the great God that you are. We pray in Jesus' name.
You encounter many problems if you try to create life in a test tube. This is just one. Amino acids are the building blocks of life, and if you artificially create them in a test tube, you get equal mixtures of their mirror images of each other. But life only uses a left-handed variety to create proteins. Origin of life experimenters have puzzled over this, trying to exploit subtle differences to purify the left-handed form, only to meet with abject failure. An average protein contains about 410 amino acids. The odds of that protein forming by chance, taking into account only this factor, is 1 out of 10 to the 123rd power. Let's quantify that. Suppose you give a snail the job of moving the entire Earth to the other side of the universe and back, one molecule at a time. He moves at a pace of 1 millimeter in a million years. Now you set up a protein factory producing random proteins at the rate of 400 tons a second. By the time one protein usable in life is produced, the snail will have completed his task. In the meantime, our protein factory has produced enough unusable proteins to fill the entire known universe. Clearly, the origin of life is supernatural in nature from an intelligent creator. The giraffe that sings. A while ago, a fellow asked us in an email why in the giraffe a nerve descends all the way to the bottom of the neck, then goes up to the other side to the larynx. Isn't that evidence that God is a poor designer? I replied, when you wire a house, do you go around the walls or do you send it the most direct route diagonally across the room? Apparently, you must be a much better designer than God, so I'll give you an experiment. Go into the giraffe and surgically shorten the left recurrent laryngeal nerve. Perhaps then the giraffe would be able to sing. Speculating on how you could design something better than God is arrogant and quite foolish.